Hey guys, in addition to the homeostasis video, we're also going to start talking about embryonic development. So there are a lot of complex processes when embryos are developing, but it's a pretty important and universal process, and it actually unites all living organisms because all living animals, for example, develop very similarly through embryonic development. So this development occurs in three main stages. The very first stage is something we call cleavage, and cleavage is the rapid mitotic cell division of a zygote, and a zygote is a fertilized egg. So that's when a sperm and an egg, or a sperm fertilizes an egg. And what we start forming is a blastula, and a blastula is a hollow ball of cells. Blast means ball, hollow ball. So it's literally a hollow ball of cells that starts forming. In mammals, the embryo at this stage we call a blastocyst. And the blastula is composed of a couple different components, but the two that are most important is this inner cell mass and the trophoblast. The inner cell mass is what's actually going to develop into the embryo. And then the trophoblast surrounds that inner cell mass, and it's going to secrete some fluids, forming a structure inside that hollow ball called a blastocele, which is just a fluid. And it's also going to form the structures that attach the embryo to the uterus, so an umbilical cord. And so just make sure that you focus on these inner cell mass and the trophoblast and the importance in cleavage. The second process is actually one of the most important processes, and this is called gastrulation. And this is when that blastula, or that hollow ball of cells, starts to rearrange all of its cells. And so we form something that's called a blastopore, which is essentially like a small opening within that blastula. And so some of the cells from the blastula will actually start moving inside of the blastopore. So the cells start migrating, and when we start getting these cells that start migrating inside of the side of the blastula, we form something called an archenteron. This is going to become the primitive gut, so that whole digestive tract. And so a key takeaway of gastrulation is the movement of cells around this blastula, and so we start forming three germ layers: ectoderm, endoderm, and mesoderm. These germ layers are stem cells that will develop into very specific organs within the embryo. So ectoderm is going to form the skin and the nervous system. Endoderm is going to form things like the lungs, liver, digestive organs, so what we call the viscera of an organism. And then mesoderm is going to become things like muscle, blood, and bones. And so gastrulation, the key takeaway, produces three germ layers as a result of cell movement and migration. Organogenesis is the third process of embryonic development, and this is really just when the embryos start developing. This diagram here is talking about how we start forming something called a neural crest, and a neural tube, which is important for a developing embryo. And this is when cells just pretty much continue to differentiate. And that's a key word that you have to be able to define. Differentiation is the process in which certain parts of DNA are activated and certain parts are not activated. That's what allows those different germ layers, ectoderm, mesoderm, and endoderm, to become very specific cells. Very early on in development, cells aren't differentiated. There's no difference between a lung cell and a heart cell. So differentiation is continuing here in organogenesis, which allows specific organs and specific structures to begin to form. Switching gears a little bit, we're going to talk about some of the factors that influence embryonic development, and this can get pretty confusing, but I'm going to try to simplify it the best I can. The first factor that will influence embryonic development is something called cytoplasmic determinants. So if you look at this diagram here, you have a sperm cell, and then you have an unfertilized egg. Well, within this egg, we have these molecules of different cytoplasmic determinants. These can be things like mRNA, proteins, etc. Well, you undergo fertilization, or the egg will undergo fertilization, and then we start getting cell division. Well, what you'll notice is that those cytoplasmic determinants, or those various molecules, are randomly separated into the two cells. That's what allows these So this plays an important role, or the cytoplasm plays an important role in embryo development, depending on which cells get which one of these molecules. The second factor that influences embryonic development is something called a gray crescent. And so a lot of eggs are what we call polar, meaning they're going to have different components of the egg. Well, there's this structure called a gray crescent, and there was an experiment done where the embryo was divided in two different directions. It was divided down the middle here, and each cell got a part of the gray crescent. And so what they observed in frog embryos were normal embryos. Well, then they cut it in such a way where only one cell got the gray crescent, and what they saw was that only one frog embryo developed and the other was abnormal. So this structure called a gray crescent influences embryonic development. The third factor that influences embryonic development is something we call embryonic induction, which is the ability of a group of cells to influence the development of other embryonic stem cells. And so this guy, Speeman here in 1924, did this really cool experiment where he took a structure called the dorsal lip of the blastopore. He had studied the dorsal lip and saw that the dorsal lip induced the formation of a neural tube, which leads to the formation of a new embryo. So he took the dorsal lip of one embryo, he put it onto another embryo, and what he saw was a new organism developing right off of the other organism. So the development of a neural fold and a neural tube happened, and then we had two embryos growing at that time. So this experiment discussed the importance of embryonic induction, or the importance of that blastopore. And then a final factor that influences embryonic development is something called apoptosis, which is programmed cell death. Constantly through embryonic development, some cells are dying off, and this is an important process that occurs. For example, early on in development, you actually have webbed hands and feet. Well, those cells that make up the webbing in between your hands and feet will actually undergo apoptosis, or programmed cell death, and hence you're not born with webbed hands and feet. However, if apoptosis is not occurring, you could have offspring that are still web-toed and webbed feet. And then the last factor that influences embryonic development, and the one that I think is most important, is something we call homeotic, homeobox, or Hox genes. So all of these are just different names for the same thing. And these are also, or more commonly known as, master regulatory genes. The best way to explain how these genes work is they essentially say, insert leg here. So in organisms, these genes actually program or control where certain limbs or where certain structures are placed. And this is just a diagram of a fly and of an embryo, and we've actually have mapped out where these Hox genes, or these master regulatory genes, are that control the formation of different structures and where they are placed. So the key takeaway is they're master regulatory genes, and they control specific locations of where specific structures are placed.